next talk is going to be by Gallant from Berkeley, and he's going to talk to us about a deep convolutional energy model for ventral stream areas beyond we do before.
I agree with virtually everything Anita said, except her last comment that VGG is the best model. We can actually build a better model than VGG, although sets or any standard convolutional deep network. But other than that, she was spot on, and all of the observations that she made, I think, are solid observations about V4.
I'm not sure your
problem.
Microphone.
My microphone is not on. Is that on?
Yeah.
Okay. So what I was saying was, this is a really big room, and I usually talk loud. I agree with everything Anita said. I think we can build a better model than a deep convolutional network of V4, but that doesn't necessarily mean we'll be able to understand them all. Thanks. I want to thank the organizers for inviting me. Parts of this talk were embedded in another talk I gave here at Rutgers a year ago, and if this seems like a rerun to you, I only have two things to say. A, the last talk I gave was incomprehensible, so if you sat through it, you probably didn't understand it, and hopefully this one will be better. And B, when they invited me, I don't know how they expected me to have done something new in a year. A year is a short time in neurophysiology. So this is neurophysiology data. I started working on area V4 in about 1988, which is 30 years ago. And I published one of the first V4 studies on shape selectivity in V4. It was actually sort of the second. Desimone and Shine had published something before, but that didn't really make that much sense. And I've been trying to figure out how V4 works ever since. So I figured I had the first actual shape-related result from V4. What I've been trying to do ever since is have the last result from V4. So a famous old scientist said there's only two papers that matter in any field, the first paper and the last paper. So I just want to point out that V4 is in the middle of the primate visual hierarchy, and that visual hierarchy is organized so as to exploit the hierarchically organized statistics of natural scenes. And we can show both in humans and in animal physiology that the layers of the visual system, the different hierarchically organized areas, match the hierarchical statistics of natural scenes. The easiest way to do that is we take an artificial network like any of the convolutional networks, like the one that Anita showed, and these hierarchical networks have a bunch of layers in them, and they're trained on a sort of generic image task, for example, image classification. Just classify what the semantic category of the main item in an image is. You show the network millions and millions of images, and it slowly learns how to categorize images. And if you look at the early levels of the network, the receptive fields that Anita showed will be very simple, and if you look at the highest levels of the network, the receptive fields will be very complicated, and that's because these networks are learning to extract the increasingly higher statistical properties of natural scenes. Those higher statistical properties, I should point out, are not known to humans. We know up to the second order properties of natural scenes, the power spectrum. We know that natural scenes are 1 over f. They have a 1 over f amplitude spectrum. And we know the third order statistics, a few of the third order statistics, but not many. But beyond that, everything's basically confusing until we get to objects, because we all know what objects are, although they're hard to quantify. At least we have a concept of what that means. So in 2014, I had a brilliant graduate student, Pulkit Agrawal, who actually was shared at that time between myself and Jitendra Malik, and Pulkit's actually now, I think, moving to MIT to start a faculty job. And he said, why don't we use these convolutional neural networks to analyze brain data? And the whole brain data that we had in my lab was from MRI. So we had a bunch of people who had watched natural scenes and watched natural movies, and Pulkit actually just fit the layers of the convolutional network to brain areas, and what he found in the visual system was that early visual areas here at the very occipital pole are in blue. That's V1, were fit by low levels of the network, and higher level visual areas that we know are semantically selective, like the parahippocampal place area that was discussed earlier today, those were fit by the high levels of this network. And I buried this paper in archive because I thought this was the stupidest, most boring result you could possibly imagine. After all, we know the human brain is hierarchically organized. We know these networks are hierarchically organized. We know one thing about both these systems: they learn by looking at the world. So of course there's going to be a relationship between these networks. But apparently I was the only one that thought this was boring, because about 15 other papers have come out showing the same thing since then, and mine never gets cited because you can't cite an unreviewed archive paper. So I learned a lesson from this, If you have a paper that you think is boring, publish it somewhere anyway. That's my lesson. All right. Anyway, the point is, V4 is in the middle. It's in the middle here. It's an intermediate visual. It should be responsible for intermediate statistics, but we don't know what those are, so it's hard to figure out what V4 does. will very briefly review the history of V4. This is the first shape-selective V4 paper. It used these weird non-Cartesian patterns because we had this lead group hypothesis that turned out to be completely wrong. But what we did discover is that V4 had a lot of neurons that were very tightly tuned for these curvature grading patterns and these intersection grading patterns. And then Ed Conner came to the lab, David Meneslis' lab, and he was a postdoc when I was a postdoc, and we argued about this result for quite a while, and Ed and I ended up with two different directions. Ed's direction was, okay, we need to start using shapes and really figure out what V4 cares about shapes. And Ed went off and started his lab and started doing that, and Anita is Ed's star graduate student, and they've been doing that for a long time, and it's been a great line of really interesting research. My attitude about this result was, holy heck, I have no way to figure out what this is, and I know that there's a bunch of other stuff going on too, and trying to figure out from first principles what is going on here is impossible, and we just have to use natural scenes. And that started a whole line of natural scene research and a big political controversy that's been going on. Well, the political controversy ended in about 2005, but the natural scenes research has been going on ever since. A few other things about these results. This is curvature tuning. This is much clearer curvature tuning from Anita and Ed. Anita and Ed also showed that these neurons are not tuned for generic curves, but for very specific curves, pieces of curvature and elements of curvature. Another group that has looked into this is Tatiana Sharpie, who's here, and John Reynolds and their colleagues, and they have looked at, tried to quantify, for example, orientation tuning, because one thing that's clear in these neurons is that orientation tuning is very complicated. There was also position invariance. This is a position invariance result from early on, showing that these gratings, these non-Cartesian gratings, have positionally invariant responses in V4. This is a more recent study from Tatiana showing position invariance in V4. Anita showed a slide that unfortunately I don't have this slide deck showing position invariance. So V4 has really complicated tuning, but not so complicated that it cares about entire objects like dogs. And it's got some moderate position invariance, but not so much position invariance as you see in IT. But more than you see in V1. It's in the middle. It's in the middle between everything, and completely incomprehensible. So I should just mention, I won't describe this result in detail, but if you give a human a V4 lesion, that human looks exactly like a monkey that has a V4 lesion. As far as we can tell, human V4 is basically indistinguishable from it. All right, so now we want to model V4. What would be a good model of V4? Well, I'm sad to say that until recently, this was the best model of V4. This was published from my lab in 2006. This is the dumbest model of V4 you could possibly imagine. This model says V4 takes the power spectrum of the images, and it creates a receptive field in the power spectral domain, and it throws away all the phase information, and that's the tuning curve of V4. And the reason we proposed this model is because we couldn't come up with anything better. And this model can sort of show different kinds of tuning in V4. For example, if you have a curvature receptive field, it shows tuning for curvature versus if you have a receptive field whose power spectral sort of tuning profile suggests that it would be sensitive to curvature. You can recover curvature tuning, you can recover various kinds of texture tuning, various things with this model. It only predicts about, say, 20% of the variance of a V4 for cell responses. And until recently, this was the best model, and we know this model is silly. So it's kind of depressing. This is another model which works well for one domain of stimuli. This is from Ed. Did you show this slide? I can't remember. This is from, oh right, because this is Ed. This is, I think, after the lab. So this is a curvature model from Ed Conner's lab that works okay if your stimuli are limited to the domain of curved contours, but fails miserably for everything else. So this is also, I would say, a modeling failure, and I'm not blaming Ed or I for this. It just turns out V4 is really hard. So about 10 years ago, I had a graduate student who was in my lab for about 9 years. Now he's at the Gallant Institute, Mike Oliver. And Mike Oliver decided to attack this problem directly. We decided that the trouble was we just could never get enough data. The typical neurophysiology experiment, you'll collect 300 stimuli worth of responses if you're lucky. And if you're trying to fit a very high parameter model, then you're never going to figure out how to fit those parameters if you don't have enough data. So we set out to collect huge amounts of data, and the way we did that was to use Utah arrays. And so we decided what we needed to do was get a neuron and maximize the total information flow through this neuron, which means we needed the entropy in the stimulus set to be as high as possible, and we decided to use natural images for that. And then we wanted the entropy in the output to be as high as possible, so we wanted to use long-term recordings more than 1 day. V4 in anesthetized animals is, since it's intermediate visual area, it's pretty sleazy, so the response to V4 are much better and more consistent in awake animals. If you have an awake animal, you can only get so many trials out of it during a day. It ends up being a lot of complications. So we wanted to record over multiple days. So instead of using these simple stimuli that I and Aoife and Ed have used, we decided to use complicated stimuli, and those were just natural movies. Animals like watching these natural movies. It's a very fun task for them. And in some experiments they were fixating, and some experiments they were free viewing. We're going to talk about the fixation data today. And then we have these Utah arrays. These are little 10 by 10 grids of electrodes that get implanted, and they never get taken out. They're just there permanently. And it turns out, by a miracle, that you can record for a very long period of time using these arrays. This was a fact that I and Jim DiCarlo discovered kind of by accident at the same time. We would bring the animal into the lab every day and show him this movie, which in this case is 450. And even though these electrodes are kind of moving around and the profile of the neuron may change a little from day to day, the pattern of responses across these movies is completely unique and diagnostic for every single neuron. So you can track neurons for weeks and weeks and weeks. So you can see in this neuron, we held for more than a month. And there's no other neuron that will have this particular peristimulus time histogram across this movie. So by showing the same 450 frames of movie every day, we could classify all of our neurons. And in this experiment, over a couple years, we collected about 800 V4 neurons. Many of these V4 neurons, we have more than 1 million frames of video. Okay? Now, I said this rule of thumb when we started this experiment, which is, if I can't figure out what V4 does with 1 million frames of video, I'm giving up and retiring. There's nothing else to be done. Because after all, this is a square root of n problem, right? If you have 1 million frames of video, to get any significant increase in the amount of signal you're going to have to model, you're going to need 5 million or 10 million frames of video, which is just insane, right? So we set million as the target. For some of these neurons, we have as many as 3 million frames of video. Okay, so now we want to model V4. We wanted to have a soup to nuts model and model as much as we could. And to do that, after many, many years of trying classical system identification methods using Volterra series and hierarchical Volterra series, all kinds of complicated things, Mike decided to go back to first principles. He would use the modern GPU-based convolutional network architectures, but he would try to essentially design in as many constraints that we know about the visual system as possible so that he could fit these convolutional networks with far fewer parameters. Now, there are a bunch of things we know about the visual system. Well, the first is that neurons in V1, the classical receptive field looks like a Gabor function, and that's because, of course, the independent components of natural images are essentially Gabors. And we know that there's 2 classes of cells in V1, simple cells and complex cells, although you can end up with intermediate cases, and the non-classical surround makes things a lot complicated. So he wanted to have an architecture that would learn simple cells and complex cells. He also built in a bunch of other things. He wanted to deal with color, so he wanted to have something that could learn LN codependency or whatever kind of color space V4 needs to learn. He wanted to have some sort of Gaussian pooling because he probably thought that would be important. He knows that the visual system takes a lot of spatial derivatives, so he thought he had a sort of a hypothesis that maybe what happens is V1 receptive fields get fed into higher levels, and instead of just pooling, there's a derivative-making operation that happens on those receptive fields. He basically tried to architect this neural network system so that it would incorporate as many of these constraints, or could incorporate as many constraints as possible. So he built basically a standard deep network. This is not what it looks like. This is schematic. There's a bunch of input. There is a layer where essentially you can think of it as taking the spatiotemporal integral, because these receptive fields live in space-time. I should point out one thing I forgot to mention. Since I'm a megalomaniac, the goal of this project was to model the V4 neurons' responses at the frame rate of the monitor, which is 16 milliseconds. So we're not trying to model the response to objects over 500 milliseconds. We want to model every single frame that happens when the animal looks at a natural movie. Right? When you can do that, then you win. So you do that, you're kind of in outer space. So this delay network in here that has essentially, we call this a deep time delay network. It has not only the current frame, but frames back in time. And it's going to learn both the spatial kernel and the temporal kernel. And then there's a bunch of feed-forward pooling levels that I'll show you in a minute. Okay, so this is the general idea. We've got some input, and then we're going to learn essentially two kinds of classes of neurons, complex cells and simple cells. I should mention, however, that these do not have to be like Gabor's. It's just that the complex cells are linked in counter phase, and the simple cells are not. And then there's some pooling levels that get happened, and then that get imposed there, and then there's some output. So this has, for 32,000 pixels, it has 10,000 parameters. This is not like a standard deep network. Remember, the deep network has more parameters than pixels. If you just use a standard deep network that Google would use, this has fewer parameters than pixels, which is what we want for a model. The features of the model aren't simply stimulus elements. They can be learned. All aspects of the model can be implemented in no neurobiology. There's nothing here that's mysterious. And the model is simple, as you'll see. This is a little more complicated interpretation of the model. There are really 4 or 5 kinds of elements that can get mixed together. For V1 and V2, we only need 2 kinds. We need this 4D input module, which is 2 dimensions of space, 1 of time and 1 of color. And we need a pooling module, where a bunch of these individual cells are pooled together to give us the output. Because we're going to model both the classical receptive field and non-classical receptive field, we need a lot of these units. There's a bunch of magic neural network food that I won't talk about here that Mike had to do to get this thing to learn, even though he's got 1 million frames in video. That's still a very high dimensional space. So Mike has some really nice orthogonalization architecture built in that orthogonalizes space and time very early on and treats the whole thing as space-time color separable, and that turns out to work really well and save us a huge amount of fitting power. That's data we can use to fit filters rather than just fitting ridiculous things that we don't care about. So these are the results. On the top is a histogram in green of our predictions. These are correlations between the model and the actual data, the responses. In blue is a standard convolutional network, which still works pretty well, but needs 10 times more parameters. In green is our network, the hierarchical convolutional energy model. And you can see that this thing does really, really well. The correlation's around 0.8 or 0.9. You might say, why isn't this perfect? There's a reason for that. This metric that we're using here is correlation. The time bins we're using here are 16 millisecond time bins. In 16 milliseconds, the most spikes you can have is about 4 spikes. That's clearly not a Gaussian distributed variable. We're using the wrong metric here. So the noise ceiling is somewhere around, say, 0.9. So basically, for V1 and V2, we're at the noise ceiling here. You can't really build a model that's any better than this. And here is our model on the vertical axis versus several other standard models. And you can see in every single case, this network just beats the heck out of everything else. Now, one thing I didn't tell you is that these V1 data came from, where is it? Oh, it's back here. Here's the receptive fields. You can see that these receptive fields, remember, these are spatiotemporal receptive fields. Here's the inhibitory regions. Here's V1. This is, I think these are excitatory receptive fields. Here's V2. You can see these all look perfectly reasonable. These came from a Utah array. So you can look at the receptive fields down here and down here. This is the luminate sulcus. You can see that the receptive fields from V1 look different than the receptive fields from V2. And there's one little fact I didn't mention here. All of these neurons came from the fovea. These are all less than 1/2 degree from the fovea. These are really tiny receptive fields. That was not intentional. That was an accident. But that's what we recorded. And normally, no one would ever record from foveal neurons in an awake animal because eye movements are on the order of 0.2 degrees, and the best eye tracker in the world can only get out in 10 minutes of arc, so you figured this would be impossible for you to track neurons in the fovea. But because we have an infinitely large data set, in other words, 1 million frames of video, we can fit these neurons even in the fovea. And a classic question people have had for 50 years now is, how do foveal neurons in V1 look compared to peripheral neurons? And the answer is, they look the same, except they're really, really tiny. But other than that, they're the same. So that makes you feel better about all the other V1 data we have. right, so now let's go to V4. For V4, the idea is we're just going to iterate this scheme. You've got a V1 population, that ends up in some sort of pooling process, and then it gets fed into V2, that ends in some pooling process, and then it gets fed into V4. Simple procedure. Anita, question?
So I'm a little bit confused about your fit then. How are you taking into account the eye position of the animal when you're fitting that data?
We have the eye position at 16 milliseconds, and it gets essentially tracked. But there's some error in it, right? Okay, there's one trick in here. You know, you think this is a pretty cool scheme. It's basically, we have one kind of module that works for V1. We use the same kind of module in V2. It's just stacked on top. Then there's the same module in V4. So basically, that cool thing about this, if you think about it from an evolutionary point of view, is you make a V1, and then you say, oh, I need another visual area. What should that visual area be like? Well, it's another V1. It's just stacked on top of V1, and it pools. And then you think, oh, what kind of visual area should be after that? Oh, it's another V1, but it's stacked on top of V2, and it's pooled. So this is a very nice, elegant architecture because you don't need to invent any new neurobiology. We already have a V1 that works really well. Lots of animals have V1. All mammals. There is one trick you need to do, though, once you get up to V4, because the receptive fields in V4 are big. And that turns out to cause a problem because of the log polar transform of V1. You remember there's a log polar transform in V1 that changes the receptive field sizes, and it turns out that if you're a V4 neuron getting its input from a big hunk of V1, and your receptive field is circular on the projection onto V1, your receptive field will not be circular in the visual field. And that was a result shown by Brad Motter in 2009 in a really elegant, nice paper. V4 receptive fields are kind of elongated toward the periphery as a result of the log polar transform. So Mike implemented the log polar transform essentially by hand at the beginning of this whole process, just to take care of that factor. All right, so now we have a V4 model that has a few more modules. It's got this 4D input again. It's got some pooling that happens. And then it has this thing called a scattering module. And the scattering module essentially implements something akin to Malot's scattering transform. The scattering transform is a mathematically nice way to introduce some degree of position invariance and tolerance to deformability while still maintaining good mathematically principled ways of combining information and preserving information. Essentially, the scattering transform is a way to get position invariance without losing frequency information, which is a critical thing you want to do as you move up the visual system. And then there's this mixing module, which essentially just does dimensionality reduction further on. So now we have the input, some levels of pooling and mixing and scattering, and they all sort of get chained together. Now, there's 172,000 input pixels because these receptive fields are big in V4, and this is only a 20,000 parameter model. So we're still satisfying this constraint of having fewer parameters than pixels. Okay. So these are the V4 results. You can see that this correlation histogram is not as good as V1 and V2, but this is V4. V4 is a lot more complicated. And at this point in the talk, I always like to point out that if you take the standard Hubel-Weasel, well, not Hubel-Weasel, the John Dogman, spatial frequency-based, simple and complex cell model, and you fit it to data from V1, you get a correlation, if you're not, you don't do anything fancy like we did, of about 0.5. And for 50 years, we've all assumed, yeah, we kind of know what V1, unless you're from Olshausen, my good friend, we kind of assume, we kind of know what V1 does, it doesn't correlate with the transform. And we've all been saying that we think we know what V1 does based on a correlation of 0.5. So my argument is if we have a correlation of 0.5 in V4, then we can use that same sleazy kind of, we kind of know what V4 does, to say that we have a model of V4. How much time do I have left? Are you giving me a dirty look? 5 minutes, all right. So if I take 15, it'll be fine. Okay, so I'll try to finish that. All right, so here are predicted receptive fields for typical V4 neuron. These are 4 V4 neurons. You can see the correlations on here. They're all really good. And now you can look at these V4 neurons and figure out what they do. Yeah, good luck with that. All of the V4 neurons are hideously complicated. A lot of them have this AM structure, where you can see there's different frequencies and different colors and different orientations of subunits all getting pooled together. And that's something we knew from Chris Baker's work, actually even back in V2. You see a lot of AM selectivity. And an AM filter is actually what you want to build if you want to break camouflage and find illusory These all are really, really complicated. Some of them, like this one over here, what is this thing in here on the upper right? This is a really pretty well predicted neuron, but you can't possibly look at any of these and figure out what they do. They just don't make any sense if you just look at the receptive fields. So this shows you receptive fields relative to the best stimuli. So, and the reason I'm showing this slide is because it shows you the input layers. Here's the V1 some V1 subunits. Here's some V2 subunits. Here's the V4 subunits. And here's the predicted optimal stimulus. Now, these V4 neurons have a lot of phase invariance, so there's not one optimal stimulus. There's a whole family, a whole manifold of optimal stimuli. These are the optimal stimuli for this V4 cell, and these are the 4 frames of the movie that elicited the largest response. Here's a different V4 neuron. Good luck figuring out what the heck this is. And look at these 4 frames of the movie. They don't really appear to be related to one another, right? That's an ongoing theme you'll see here. Here's another V4 neuron. This is really, looks like a bird of paradise or something. And here are 4 frames that maybe have something in common, kind of hard to tell.
Jack,
Yes?
can you give us a sense of how much these V4 neurons are responding to those frames? What's the firing rate for those frames like?
I can't remember offhand, but we have 1 million frames of video, so these are really good stimuli.
Well, not necessarily. They're bad.
They're better than if we just pulled them out of our posterior, right? I mean, we've got 1 million frames of video.
Okay, so you can't tell me what the best response to those stimuli
Not offhand, no. But it's almost certainly higher than if I picked 100 stimuli at random, right?
Yeah, just pull the answer out of your posterior.
I do that far too often as it is. I don't know the peak responses, but the most effective stimuli in these 1 million frames of video are generally pretty high. Right? All right, so now this is what Mike did to try to get a sense of the relationship between the best stimuli and the receptive field. So what he does is he starts with the best stimulus for a neuron, and then he essentially drives down the gradient to try to increase the cell's response more and more and more and more and more, and find the best possible stimulus based on the best stimulus that we started with. Right? So you'll see that we're slowly transforming between the best stimulus, blue thing here, and the best stimulus that we expect would occur if we had actually shown it. You'll notice a lot of these V4 neurons are responding at frame transitions, and that's really common because every time you move your eyes, or every time there's a scene cut in a movie, every neuron in V1 gives a huge phasic response, and that signal percolates upstream. It's like a lock-in amplifier. It's a resetting signal that the system uses to figure out that you moved your eyes. And it has a big effect on the spatiotemporal responses of V4 neurons. So you can see that these receptive fields don't bear any obvious relationship to anything you could actually make sense of, but that they are clearly related to stuff in the scenes. It's just not stuff we can talk about. So now we have 800 neurons. We have 20,000 parameters to this model, and we have to do cluster analysis. That's impossible. If we want to figure out what the clustering is of the neurons or what the dimensions are, it's very, very hard because the parameters of this model is 20,000 parameters. We only have 800 samples and 20,000 parameters. So there are some tricks you can do to try to cluster your data in situations such as this. And the first trick you do is you squint really hard at your data and make up a story. And that's what Mike did here. He made up a story. He decided, and I bet other people in the lab separately go through the data, and they also think this is a good story. So we're going to say there's contour cells, there's curvature-ish cells, there's temporal contrast cells, and there's AM cells. But of course, as I said, this is a made-up story, and this is all a continuum as an intermediate. So now Anita brought up a valid point, which is, wait, you can do this with a trained network, as I said at the beginning of this talk. The only difference is this trained network is going to have 10 times the number of parameters as our architected network. But still, it will fit. So let's pit this pre-trained network, where we select features, against the architected network, where we fit the features. And if your correlation is below about, say, 0.45 or 0.5, it's kind of 50-50 which one of these you want to pick. You can either pick the artificial network, which is on the horizontal axis here, or our network. They both kind of work about the same. But for the neurons, we can predict really well. You can see that for those neurons, our network always works better than the pre-trained network. And we have 10 times fewer parameters here than the pre-trained. So if you ever want to actually figure out what the neurons do, you want as few parameters as possible. There's another cool game you can play that I'll just mention, which is once you have these neurons, you can do an in silico experiment. This is something we first did back in 2011 in our MT paper, where we used natural images to fit models of MT and ended up with the best, as far as I can tell, still the best model of MT out there, which is a spatiotemporal energy model from Shinji Nishimoto. And what Shinji showed in the appendix of that paper is if you take classical MT stimuli, like PLADs, you can replicate any effect anybody's ever reported in MT based on this model that was trained on natural images. Same thing in V4. We have a model trained on natural images, and you can replicate all the kinds of phenomena that people have reported in V4. For example, this is a non-Cartesian tuned cell. This is a little pointy, I forget what we're calling these, a bunky tuned cell beneath an head. And I just want to point out that this idea of taking a certain kind of representation, if you want to iterating it is not a new idea. One of my favorite all-time computational neuroscience papers is from Jan Karpman and Mike Lewicki, who basically took Bruno Olshausen's and David Field's V1 model and essentially iterated it one on top of another and found receptive fields that looked remarkably, actually, although they used a different scheme here to show them, looked remarkably like our receptive fields. So I think that's all. Let's skip back then. So we propose a hierarchy really, I can't even see what this says. I'm gonna claim that we solved V4. Now, obviously we didn't solve V4, but I'm gonna claim we solved V4 as much as the classic people solved V1. The problem is, by solve, I mean I fit a model that predicts really well. I don't mean I actually understand what V4 does. I actually have no idea what V4 does. It does everything. V4 is right in the middle there. All the visual input that goes down in the infratemporal cortex in monkeys and primates and humans goes through V4, and it does all of the stuff it needs to do to, at this intermediate stage, to pass information down to the next level. The problem is that stuff, I would argue, is not human interpretable. I mean, you can find V4 receptive fields that are tuned for this segment of space, which involves my shoulder, my head, my shirt, and the blackboard behind me. And that is not a kind of thing that we have any language to talk about. So, you know, we can talk about really simple features in images like edges and colors. We can talk about whole objects in images because we have names for them, like, you know, grandma. But the stuff in the middle, we don't really have names for it. And as a consequence, we don't really have any intuitive access to it. It just doesn't make sense. They're hideously complicated receptive fields, but they're there. They're receptive. Anyway, that's it. Thanks for your time.
Time again for questions.
Yeah, so Jack, can you clarify something about how you dealt with the scanning of the monkey? Were they looking around, were they fixating? That's part A. And part B, if they're looking around, you could do your analysis in different time slices relative to the onset of the saccade and also its direction. So what's the outcome of these?
Thanks for asking, Allie. We have all these data come from a fixating animal. So the only saccades you have to worry about are microsaccades. So this comes from epochs that are about 5 seconds long, and then they'll break fixation, and then they have to reacquire, and you run another 5 seconds, and you stitch that all together. We have an enormous data set from free viewing. Because what ends up happening in these kinds of experiments is eventually your guy says, I don't really care about doing this fixation task anymore, so you say, okay, we'll just watch the movie. So we have tons of hours of these guys watching movies, and we haven't processed that data yet. Just to mention how horrible this kind of data is to process, it took Mike over 2 years just to cluster cut the neurons and extract the individual neurons from these and stitch them together across days. I mean, that process was really 2 years of just horrible, horrible work. So it takes a really long time to basically go from the raw data that's taken across days when neurons are coming in and out at any time during a day and to make sense of what neurons go together and make sure you haven't made any mistakes before you can go to this other problem. So we just have that again.
One more question? Okay, you know.
error.
checked out of school. Can you tell us, I lost track of the numbers, that input space has what dimensionality relative to the output space of the V4 model?
And I think for V4, so what happens for V4, I think these are like typical numbers because everything got scaled to 3 times some receptive field measure. But a typical reduction would be like 130,000 pixels down to about 20,000 parameters for V4.
And can you get any sense of what that represented space looks like, for example, by drawing samples from the set of images that would
Right,
responses?
So correct me if I'm wrong, I think your point is, look, yeah, there may be 20,000 free parameters, but it's doubtful that V4 neurons as a population span this whole 20,000 parameter space uniformly. It's probably likely that some of these parameters have a lot of bias. And if we could recover that across the population, we could find the manifold that all the V4 lives on and actually try to reconstruct, essentially by aggregation, the tuning space that is represented in V4. And is that what you're asking about? Great. Do you want to do that? Yeah, that's it.
That and exploring it by drawing samples.
Yeah. We're working on that in several different directions. That's a really hard problem, but a really, really important problem. And I personally will not be satisfied till we solve that. You could imagine, for example, there's multiple ways to draw samples. You could draw samples of individual neurons, which is what I did here. You could basically take the parts of neurons and recombine them into plausible new neurons, right? There's a lot of ways you could do resampling to get at these issues, and we have just begun to explore that. It's a really important problem. I suspect what we're going to do, to be honest, given the manpower I have, we're probably just going to put these inner neurons online and let people go to town. So I'm not sure when that'll happen, but I wouldn't be surprised if this data set's made available earlier rather than later.
Jack, can I ask you one final question?
Yes.
The filters you create are spatial, they don't should be spatio-temporal.
No, they're spatiotemporal.
But where's the dynamics in the temporal?
So the way to think about it, this isn't exactly what happens, but the way to think about it is that this deep time delay network, so usually when you're essentially doing system identification on neurons or voxels, and you have to deal with time, the easiest way to deal with time is to essentially take your current spatial frame, and vectorize that, so now I have a vector, but then I take the frame before, and I vectorize that, and I stick it before. I take the frame before, and I stick it before, and I stick it before. And so now basically it's fitting this long spatiotemporal vector, right? And then when you want to look at it, you unravel it and put it all back together again. Now, that is not exactly what we did here because there's 130,000 pixels and 12 time delays, so that would be, over 1 million pixels you'd have to deal with, and that's an impossible fitting problem. So that's where the magic juice comes in that Mike spent years developing these really spiffy, fast, efficient ways of rearranging the data and marginalizing out things you can marginalize out and getting rid of interactions, making things separable when you can make them separable in order to reduce down to 1 million input pixels to something smaller. And that's actually why Mike is at the Allen Institute now, because the Allen Institute had a problem, which is they had a bunch of data from mice, and they had not been tracking the mice's eyes. Turns out mice move their eyes. If you're doing visual experiments and you didn't track their eyes, you have a serious problem, because you spent a lot of money to collect a lot of data that you can't use. And Mike figured out a way to take this model and add on a component that learned where the mouse's eyes were. By basically taking advantage of the fact that, eyes are continuous, right? And so basically he could recover the eye tracker information from the mice that they didn't actually record, and showed that when you did that, the receptive fields went from being nothing to being actual receptive fields in mice. And they decided that was worth giving the job. I think that was a good decision on their part. So Mike's doing that now. So you can do a lot with these networks.
Okay, thank you very much. Now,
Thanks, guys.
Sarah.